Well, thank you very much, everyone, for coming. You've probably all had lunch, so you're looking for a nice, quiet session to fall asleep to. We're going to make sure, hopefully, that this is lively enough to move you through that postprandial dip. I'm very pleased to be able to bring this discussion to Groundswell, because several years ago, one of the very first occasions I came here, I was encouraged to come because someone thought it would be something I was interested in, and I walked around being a sponge, just soaking up all sorts of things. I'm not a farmer, and I had an awful lot to learn. And I went away at the end of that, and sort of a penny had dropped, that the way this new farming approach was dependent upon, and the role that glyphosate played in this new regenerative approach really hit home to me, especially as there was a lot of chatter at the same time in the media and other places of the EU was at the time considering banning glyphosate, and it became apparent to me as I was driving back down to Devon, or wherever it was I was living at the time, that if that was to happen, an awful lot of the good that was going on here would become practically impossible very quickly, and people may well give up and go back to ploughing. And so that set me off on a lot of thinking, and then I was very lucky to be introduced to the two colleagues with me on stage, who I hope I can both call you both friends now. I hope, because we've been
steady. That's pushing it a bit too far, Andy.
I'd like to think.
Let's keep it that way,
as
Andy.
friends. First, the introduction was great, but secondly, for you to also be interested in allowing us to come and do some research on your soils was it was really exciting to be able to learn from you. And what we want to do today is share our experiences, and that's all we want to do. We're not here to preach. We're not here to tell you what to do. We're here to help you learn from our experiences and make your own informed decisions. And that's really what I think we want to do. We're all clear that today we're not talking about desiccating crops. We think that's an inappropriate use of glyphosate, because we don't have GMO crops that are Roundup resistant. We're not talking about using multiple applications throughout the course of the year as a weed control. We're simply discussing the use of glyphosate as a means to preparing a seed bed or clearing the land for the next crop. So that's really the framework that we're talking about. So, hello, thank you everyone for coming. My name's Andy Neal. I'm a soil microbiologist at Rothamsted Research. And everything I've learned about soil tells me that one of the most precious things that soil has that we need to think about protecting and growing is the microscopic pore networks in soil where microbes live. It's important because that's what regulates oxygen movement and water availability, and completely dictates the way microbes respire in soil. And ideally, a porous soil which has lots of organic matter in it will have a lot of oxygen availability. And that's very important when we think about glyphosate, because if you think about the chemical structure, we know pretty much generally that the sort of breakdown pathways of glyphosate in soil. And a key part of that is breaking this carbon-phosphorus bond in glyphosate. It's quite a rare bond in nature. You don't find carbon-phosphorus bonds very regularly in nature. It's also an extremely energetically demanding bond to break, which means if microbes are going to break down glyphosate very efficiently and quickly, they require a lot of oxygen. So my thinking upon leaving here and that first experience was soils need to be in a good, healthy state to be able to deal with glyphosate very effectively and effectively turn it over very rapidly so it doesn't persist. So I was very interested in understanding how tillage disrupts this very important pathway, a very important network that is allowing oxygen availability to be sufficient to break down glyphosate effectively. Now, even if we were all to agree that glyphosate is a wonderful thing and we should all be using it, there are still very good reasons why we wouldn't want to use it injudiciously. And that's really because we will always eventually see resistance appear to glyphosate. And right now, we already have reports of glyphosate-resistant weeds in the UK. We need to be using it very carefully if that's the decision we make. And so that's the sort of context of what we want to talk about today. Now, I'm going to pass over first to Ian, and I'm going to allow you all to introduce yourselves, because I know I may think of you as friends. I'm sure you would rather introduce yourselves. So first of all, I would like to hand over to Ian Wilkinson, who's going to talk about his philosophy of what they're doing at Farm Ed. And then we're going to hear from Tim Parton about what he's doing and the way he uses glyphosate in his management. In the middle, we're going to have some scientific data that we have collected, that Rothamsted has collected on the soils of the two farms. And then we're going to sort of wrap up with, I suppose, a position of where we think we need to be. So I'm going to shut up now, Ian, and I hand over to you.
Thanks very much, Andy. Thanks everyone for coming to the session. We knew it would get attention, obviously, and it's a bit daunting sitting up here talking to you guys because every one of you is going to have an opinion on this. We know that, but we really want you to engage with this. So please feel free to say whatever you like, and I hope we will be able to do the same thing. So some of you will know me. I've sat in these tents before representing Cotswold Seeds, which is a business I've been involved with for 40 years. And so I know a little bit about grass seeds on farm use. So that's really where my knowledge comes from. So I'm a fanatic about forage legumes in farming systems. I'm a fanatic about crop rotation. I really don't like simple farming rotations. I'll just put it out there now, because if you're wondering why I'm here. And I think also, when I was a kid, we were using Roundup. Roundup came the year Cotswold Seeds started, which was 1974. And to Andy's point, this is not the place to get into this straight away, but I would just like to say, I'm 62, it's been with me all my life, and we have become dependent on it. And whether we agree with that or not, that is the reality. So we are able to question our long-term use of any product. Think of antibiotics. We know that if we do the same thing every time, we're going to end up potentially in trouble. And that would be the same with growing crops without rotation, with antibiotics, anthelmintics, you name it, same problem. And I believe we potentially have the same trouble here. Ploughing has been demonized. Unlike Andy, I actually quite like ploughing, as he knows. I'm one of those people that didn't sell the plough and have kept it. Andy's mentioned that we have resistance to weeds, and if that sort of thing continues, then we will have to use physical cultivation to destroy weeds, because lasers don't seem to work yet, and I don't know of any other technique that's commonplace that will do it. So we know there's environmental effects. We are being surrounded by information that, particularly with water and pollution to water and the effect that's having. It's probably not the place to talk about it, but these are the reasons why I feel passionate about it. We know that it's having perhaps positive and negative effects on soils because of the way we're using it. And I know Tim's going to talk a little bit about toxicity and potential human health, but I do worry about it. And I'll just finish by saying on this section that I'm just concerned that for two reasons. One, I have my urine measured and I contain 1.42 nanograms of glyphosate. And if I do, you probably do too. I have no idea whether that's a problem, but I'm just saying it's out there. It's in us, it's in the environment. And right now, we've got another 8 years until the regulation that governs its use in the UK is up. 8 years isn't long. That's the crop rotation. So I feel like we need to be thinking about alternatives now. So on that basis, very briefly, this is Farm Ed. So I've been there 12 years. This is a photograph of a helicopter photo before drones of the farm. And this was very much part of the Cotswold farming scene, Cotswold Brash. And on top of the farm, it's a small farm. On top of the farm has been, since 1953, continuous cereal cropping. So when we bought the farm, we had the ideal place to do an experiment, which, thank goodness, Andy and other colleagues said, you should keep a control plot, which we did, because otherwise we'd have lost all this. So if you are thinking of changing your farming system, at the very least, please keep something back to measure against to see how your changes are progressing. Anyway, this is the farm. Oolitic limestone, West Oxfordshire, north end of the Cotswolds, typical arable fields. These are actually quite small ones. They're at 17 acres each, and most fields around us be 40, 50 acres now. So anyway, we got the control plot. We kept a bit of that, and it had either been ploughed. In fact, it had been ploughed every year, often twice if there was a spring crop coming up. So it'd be ploughed one way, you hate this, Andy, but the ploughman really enjoyed it. I mean, he did. I mean, Jim who used to plough this land, he loved it, and so did his wife, Wendy, and if one wasn't on the tractor plough and the other one was. And if you think back to those days, to get a seed bed, to get it, broken down over the winter, to get a seed bed for the spring barley crop or whatever was going in, that's what you did. So that was quite normal practice. So, lovely control plot, and it has had glyphosate on and off for all those, well, not all, but nearly all those years. So we, in 2013, took all the regular tests. We did loss on ignition tests, of course, and DOMAS. Soil organic matter, which no doubt we might discuss in this conversation this afternoon, was about 4% across the top of the farm, very consistent. So that was with heavy cultivation, as part of the system. So the demo farm now, and those of you that have been, I know some of you have, we've basically fragmented it all down, and we've got one plot of about 1 hectare, which is still using the same system. So continuous cereal crops with glyphosate usage, and we've now had 3 years of no-till. And I know you're all thinking, well, that's not what I would do, because, of course, we wouldn't have a continual crop of wheat, would we? But that's what we're doing, because that's what the card we were dealt when we went to the farm. So we didn't want to lose all that data, so we kept it a bit like Andy's Rothamsted experiments. These long-term data sets are going to be really important in the future. And then next to it, we thought, well, we know loads of other people are doing great work, and Tim's work, for example, is a really good solution. What we wanted to show people, particularly those people that weren't aware of the challenges, maybe people that are coming to agriculture new, or people that are coming to crop rotation new, mixed farming, diverse farming. What does it look like if you put a lay into the rotation? So we're still growing cereal crops, but in fact, just over half of our 8-year rotation is fertility building. We're not organic, but we choose not to put any synthetic products, and I'm choosing my words carefully, not to put any synthetic products. So that includes all of the pesticides, including glyphosate, and it also includes fertilizers. So we've set ourselves a hard task. We literally went cold turkey on 8 strips in an 8-year rotation. And what's important to recognise is that we're going to be talking about some of the changes that have happened to the soil between these two systems as we go through this next hour. And so that's why we've got the system. And I know that each of you would have done it differently, but we wanted poles apart. We wanted no-till or minimal till or ploughing and glyphosate, continuous cereal, and then contrasting that with a highly diverse rotation. Let's see what the differences are. That's the same shot then from the in the air. You can see it looks pretty fragmented, which is a whole nother story, but that's what we've got. And it's been really interesting. A lot of people, Andy has very kindly been sampling from these fields, and I think there may even be 1 or 2 people in this room this afternoon that have been doing the same thing. And please feel free to chip in, please. I can't see you, but if you are there, thank you. And there's others I know, sure. So we will get a mic to you if you would like to chip into this, because there's some other scientists in the room that would be able to contribute. That's what it looks a little bit close up. So we've been in a stewardship scheme on this, which is economically absolutely great, better than the other crop actually of monoculture. But that's what ours looks like. We've just come out of stewardship into SFI, and the only thing we've dropped is the bird mix in favour of another cereal. I think it's over to Tim.
Gosh, that was a bit of a shock, wasn't it? I was going to sleep there. So I'm Tim Parton. I'm a farm manager in South Staffordshire. Where I farm as... Will you stop laughing? Where I farm as biologically and nutritionally as possible. I'm a big believer if I get that plant balanced, it won't get sick. So I just use nutrition and biology. So I don't use fungicides, I don't use insecticides, I don't use growth regulators, I don't use pre-emergence herbicides. I still use glyphosate because I still feel it's the best thing. When we came to do this trial, Andy asked me to plough a bit, and I nearly fainted. No way you're bloody ploughing my land. I've worked that hard to get everything back. I just wanted Andy's help to prove that it was the right thing to do. The picture that we're looking at there, that was a crop of lupins, and that was crimper rolled, and I drilled the lupins, so I kept the soil covered. By keeping soil covered, we're not going to get the weeds or retain moisture. And we're getting over the problem of having to need a herbicide in my mind. I do grow crops with no herbicide use at all. If I can have the right weather conditions, I can control the cover crop without glyphosate, and I don't use any herbicides at all. But in our maritime climate, that is quite difficult to do. Unless anybody's got that crystal ball that can control the weather, we're always up against it. Having glyphosate in the cupboard is fantastic. I'm totally against it going on to a crop that's going to go into the food system. That just seems madness to me. We're a living organism. It's going to have an effect on our bodies, and I personally don't want to eat it. So I don't want to sell any food and expect somebody else to eat what I don't want to eat myself. I always use the analogy, if there was 2 plates of sandwiches in the room and one had been grown by me and it had just had the nutrition on it, and one had got all the active ingredients of all the fungicides, all the herbicides, all the insecticides, all the growth regulators. I think I know which plate of sandwiches most people in this room would be queuing up to take. So I'm never expecting anybody to eat food that I won't eat myself. That was the start of the trial. So we did it in 4 sections. One just using tillage. I did disc down to 4 inches to have tillage, just to keep Andy happy and just as we could start to have a comparison. And then we did 2 different types of glyphosate, and one tillage with no glyphosate, tillage with glyphosate. When I'm using glyphosate, I always steward myself, so I use citric acid, which enables me to lower the pH of the water. I can then reduce the amount of glyphosate I'm using. I'll always use fulvic acid. Again, it's got a cation exchange capacity of about 1400, so again, I can reduce the amount of glyphosate I'm using. And then I'll always add some molasses in there. The molasses is to stimulate biology. So any glyphosate that hits the soil, the biology will eat it and degrade it, so it's not there. And the trial showed with Andy's work that after 2 weeks, wasn't it, Andy?
Well, if you just kind of wait until we get onto the slide, we can do it properly.
Okay.
It's like,
Mardos.
you're giving the story away now.
Mardos. But the one thing you can see from the slide there, where I didn't use any glyphosate and it was just tillage, it was a very dirty crop. And you can see on the one side of the picture there, that carried right the way on through. And I didn't harvest it at all. We did combine it, but there was nothing there. The weeds had overcome all the lupins and there was nothing there to harvest. Where I'd got my cover there, where I'd crimper rolled, I had a good crop of lupins, which was just proving a point. If I can crimper roll and do it right, I don't need to use glyphosate. I don't want to use it, but I want it there. I'm old enough to remember the problems we used to have with couch grass. Ploughing doesn't control couch grass. We used to have to cultivate it. We'd rake it into a pile. We'd burn it. It's bloody hard work. There's no way I want to go back to that. Well, I wouldn't be able to do it now, but it's still there. If we don't have glyphosate in the arsenal, we're going to have problems in my mind. Once you get a healthy soil, we won't have the problems with couch, but we need something to help us if we do have a problem. Soil is a living entity. Everything that we do to soil has got side effects. It's just choosing which one is the least damage to keep that soil alive and keep it working, which is why I'm always about keeping the biology there and keeping that soil working so as we can get that soil functioning, that we have all the nutrients available and we don't need to use all the synthetic nutrients that we've been applying. Because to me, as with synthetic usage has gone like that, human health has gone like that. You don't have to be a rocket science to see it. When I was a kid, we didn't have all these Alzheimer's and autism that we're having now. It's all becoming a major problem. Cancer's a regular, everyday occurrence. Nobody seems to ask why are we having all these problems, what's causing it. But we've got to start and ask those questions. And I read a paper the other day about glyphosate, and it's probably the 3rd and 4th generation where they, in mice, where they saw the problems of the mutations happening to the internal organs that were going to cause us problems. We shouldn't be feeding all this poison to our children and to the planet moving forward.
No, I've got my own.
I'll be quiet now.
So that's one of my learning points. If there's any other scientists in the room, make sure you know who you're talking to before you say something stupid like, "Would you plough half the field for me, please?" So that was a very interesting discussion we had.
You were nearly kicked off the farm.
Now, when we worked on Tim's farm, I worked with colleagues at the University of Nottingham, particularly interested because I talked about this soil poor network, and we visualized that using X-ray computed tomography and with colleagues at the University of Nottingham. And I've got some examples here from Tim's fields. And in these images, the pore spaces are in black, and the soil itself is the white space there. And what I'd like to really point out very quickly is that Tim's before cultivation, you can see there's some nice pore space in here. There's some root and worm pores. Upon cultivation, it kind of fluffed up the whole soil structure, particularly in the surface here. You did a 10 centimeter deep cultivation, and you can see the results of that. It's incorporated an awful lot more of pore space in that upper layer. But 7 weeks after that cultivation, I'm sorry if I'm standing in the way, 7 weeks after that cultivation, all that fluffy material collapsed on itself. So, as you say, nothing that you do to soil is without a consequence. And the other part of what we really wanted to look at was how long is glyphosate persisting in these soils? And harking back to my original hypothesis that oxygen was going to be very important in this, what we observed is in the cultivated soils where for at least 1 to 2 weeks we had this very fluffy texture with lots of oxygen in there, glyphosate was below detection limits. So again, we did this with the University of Nottingham. Glyphosate was below detection limits within about 12 days. In the absence of that cultivation, where we have a less pore space, less oxygen moving around in that system, the persistence of glyphosate was slightly longer, out to approaching 30 days. So clearly the amount of oxygen you're incorporating, the management of how you're preparing the soil for receiving glyphosate has a clear effect upon how long that material is in the soil before it gets degraded. Now, we also did X-ray computed tomography on Ian's soils as well, and there's slightly different approaches, and they're presented slightly differently here. Here, the pore space is represented as a heat map with the smallest pores down at 100 microns, which is where I start getting really excited, all the way up to 7 millimeters. I can see there's a heat map representation. Now, that you didn't talk about it, Ian, but you have a permanent pasture that's been pasture for many decades and has a beautiful, very diverse pore space. You can see evidence of roots here at the surface. But the point I want to get across is down to a very decent depth in these soils. There's a lot of pore space and a lot of diversity of different pore sizes. The herbal lays, which was in its 4th year when we measured it, also has a good distribution of different pore sizes, and they're all very well connected as well. And we can't go into why connectivity is cool, but connectivity is cool when you're thinking of pores. The no-till cereal also has a reasonable distribution of pore sizes. Now, what we were really shocked by was when we looked at what was happening in the wheat rotation. So this is the 4-year herbal lay that was ploughed, and we took these samples 6 weeks after they'd been ploughed. The loss of soil structure is not too much of an exaggeration to say quite catastrophic, in the sense you've lost a lot of pore space, you've lost a lot of that diversity of pore space. Now, the likely consequence of this loss of pore space is that water is going to behave and be stored and will move very differently, and of course oxygen will move very differently through these soil profiles as well. So we were very interested to understand what was going on with regards to soil microbiology and, of course, greenhouse gas emissions. These are much finer resolution. So these are soil cores. These are 2 millimeter sized aggregates. And although we're still in the process of analyzing these, you can say that even at these very fine pore scales, where we're looking now down into hundreds, down to 1 or 2 microns, that the management has had a huge effect upon the abundance, the distribution, and what we hope to be able to show, the connectivity between these pores. All of this influences to a great degree the way oxygen and water moves through these and the availability of oxygen and water to organisms living in these soils. And I have very briefly just some data that we collected on Ian's fields. Ian very quietly took me aside and said, you really don't want to put too much scientific jargon into these. So what I've done in each of these boxes, I've provided a very simple statement of whether there's differences or not. I'd like to introduce you to something called an effect size that really says how important the treatments have been in causing the differences we're seeing in the data. So if you have a perfect experiment, you'd have an effect size of 1, where all the variability you're measuring is due to your treatment. If you have a lousy experiment or a not particularly strong treatment, it would be down towards 0, where it's having no effect upon the data. So everything has an effect, but we need to understand what's having a greater and lesser extent effect if we want to try and understand how to manage things better. So the point I want to make here is that, as you might imagine, the permanent pasture has good organic carbon stocks, as you might expect it to have. The herbal lays are doing very well. But when we looked at the carbon stocks in the no-till cereal plots and then the plots that had been ploughed to go into wheat, there was actually no difference in the organic carbon stocks. So there was a large loss upon 6 weeks, or within 6 weeks, a large loss of carbon from those soils that had been herbal lay that were ploughed. So ploughing does have this well-acknowledged influence upon soil organic carbon stocks. So it's really changing the way energy is moving through the whole system. Microbes rely on that energy as carbon to metabolize and build biomass and do all the wonderful things that we want them to do. At the same time, solar microbial biomass, which we were measuring by measuring PLFAs, I'm sure there's plenty of stands here talking about using PLFAs to look at soil health, we saw a very similar response, as you would expect. But the effect size upon the soil biomass of these treatments was larger upon the soil microbial biomass than it was upon the soil organic carbon stocks. So those microbial communities are much more sensitive to management than the stocks themselves. And as you would imagine, if you consider the total number of bacteria or the total biomass of fungi, they were also changing. So really just to say that it was the whole biomass of the whole system that was being changed by this management. And ploughing appears to have a very strong influence upon that, because whatever we were measuring in the herbal lays was reduced quite considerably when we were looking at the soil that had been ploughed only 6 weeks before. And the other interesting thing about using lipids is that the lipids are very dynamic in a microbial membrane, and they adjust them all the time to maintain the lipid function, and particularly in response to changes in the environment. So there's some evidence that we can use them quite effectively to look at what stresses microbes are experiencing in soil. And we've got 3 examples here. One was particularly high in the no-till wheat, which is a replacement of lipids that typically occurs in very hot and dry soils. So it's actually a marker for a group of organisms called actinobacteria, which thrive in hot, dry soils. So there's evidence that when you have not very much soil cover, a lot of insulation on there, and the soil temperature goes up, we can detect that in the lipids we're measuring in the soil. So that's one stress. The next is starvation. We know that microbes that are deficient in energy will replace certain lipids with other ones to try and be more efficient in the way they're using carbon. And we saw that, as you remember, these 2 treatments had the lowest amounts of soil organic carbon stock. That gives us the highest soil starvation response in the microbes. Finally, there's a stress response that we know is particularly sensitive to changes in pH. Now, you have a 2-unit pH change across your farm. High in the top of the farm, very low down in the pasture. And what we saw is that there's a very strong stress response due to that pH change in the pasture. Now, I'm not just showing you this because I think it's really cool. I'm showing this because if you look at the effect sizes of all these, the biggest one we measured on your farm was not due to glyphosate. It was due to the change in pH across the farm. So the soil microbiology seems to be very sensitive to environmental stress. But often those environmental stresses are causing much changes in pH, which are giving a much more potent biomarker response than any of the other managements that you're doing. So I think it's really important that we think, when we think of using and how we use glyphosate, that there's a context within which we're using, and other things are also influencing the microbial communities. In this case, pH was very important. And finally, we were very interested in understanding the nitrous oxide emission potential, because the other thing that happens when you don't have very much oxygen in your soil is microbes respire anaerobically. So a lot of the nitrate that you might make available in those soils ends up being hijacked and used for respiratory purposes and turned into nitrous oxide. So we took some intact cores back to the lab and tested the nitrous oxide emissions from soils at different water potentials. The reason we did that is because water controls how oxygen moves through soils, so you would naturally expect wetter soils to emit larger nitrous oxide amounts of nitrous oxide. In this case, what we saw, and bearing in mind the difference in structures we've just talked about, if we really just concentrate on the no-till cereal and the wheat rotation, the no-till cereal was releasing about 1/3 of the total amount of nitrous oxide that your tilled wheat was. That's a big difference. That's a lot of nitrogen being lost from the plants, but also a large amount of greenhouse gas going into the atmosphere as a consequence of the management. The other thing I just want to point out, I think this is the final thing, is that if we look at the range of water-filled pore space that nitrous oxide is released from, the wheat rotation, so the tilled wheat that up until 6 weeks before had been herbal ley, was already emitting nitrous oxide at about 40% water-filled pore space. That's actually quite a dry soil. So that suggests a lot of that soil, although appearing dry, a lot of it was actually anaerobic at that time. The no-till cereal didn't start emitting nitrous oxide until it was about 60% water-filled pore space. So the whole dynamics of nitrogen in these systems, which is dependent upon the availability of oxygen, which in turn is dependent upon that structure, is changing in response to those management decisions. So just to summarize that, do I even need to go over that? Like cultivation, more oxygen, we see glyphosate is degraded more rapidly. You have a lovely benefit of herbal ley, increases the soil organic carbon. But I think we've agreed is critically how you get out of that very nice system into the next phase is becoming the most crucial part of this discussion, I think. So on to your big question. Thanks,
Andy. May I? So as you can see, it's really simple, isn't it? So, you know, I've been given that information ahead of time, and I've been able to digest it slowly. And I think, you know, for an agricultural apprentice going to agricultural college for 3 years, learning study farm and grassland management, and then being, you know, running a seed business and now having a small demonstration farm, I'm not really the right person to comment on the results. But what I have got from this is that, you know, it's complex and it depends on how you look at it. You know, we have, like all of you in the room, we've been measuring lots of things, including soil organic carbon, and the results are not consistent. But what I think is consistent is the fact we can build soils in rotation. And I think, as Andy said, the big question is, what do you do when you've grown your, well, in my case, it's the herbal ley, 4 years, then we terminate it. And I put an image here of the crimper roller on. So we tried a crimper roller. We made one like Rodale did. I think, Tim, you've done a similar thing with Cambridge rolls, or perhaps even a heavy roll.
Tim has a movie in a minute.
Okay, sorry. Then but the thing is, we couldn't make it work. And I tell you for sure, you can't make it work on perennial crops. You know, because the herbal ley, if that's where the fertility is being built in our arable rotations, and if that's what, you know, look, the cows are out there now and to your left, and that's going to be arable cropping, I'm assuming. That's the really good stuff that's doing all this hard work. And then if we then destroy it, so there's a couple of things I want to say about destruction. One, when we're measuring this stuff, we really should be measuring a metre down, not 30 centimetres or 40 centimetres. If you've tried digging into soil to measure it and getting a representative sample across the field, it's really, really difficult to do it. And I am of the belief there's a lot more going on below the surface of the soil than there is on the top. And I think we need to think in our heads about how much stuff we're leaving on the top when we're doing no-till systems and how much stuff is being mixed in with the plough and so on. There's a lot to this that we're perhaps ignoring. I also think that there's a big difference between real-time in-field experiments. And the great thing about what Andy's done is that he's taken the stuff back to the lab. And I know we've got possibly a soil scientist in the room that's been doing a similar thing. And also we've been measuring in the field. And again, those results are different. So if we're measuring nitrous oxide emissions on our fertilised control plot of wheat, which we have been measuring, and if we measure it just after the fertiliser's been applied, guess what? That's where all our N2O emissions are. So you've really got to, and I think for me, that I think Gabe Brown, I caught only a tail end of his thing, but so I think he said something along the lines of, you know, we all know there's context, the sixth principle, and now I think there's focus. Am I right? Is that one that I think he was been mentioning recently? And focus is really drilling down into your own situation and focusing on what you're trying to do with these soils. And I think he's right, obviously, but you know, it isn't as simple as just looking at one element, and I think Andy's shown that. So how do we terminate? In our case here, we know we've got a lot of carbon below the soil. We don't want to be ploughing up. These are Cotswold soils, and look, just below my nose, we're into really stony stuff, and the last thing we want to do is to be bringing that stony stuff up onto the top. If I used a sprayer and put glyphosate on that, I'd probably be needing 4 litres a hectare. And I'm sure that that's not what you'd be using, Tim, on any cover crops or anything. Because, if we use glyphosate on the herbal lay, that's a lot of active ingredient going into our environment. There's something like 900 million tonnes of glyphosate used worldwide. Can you believe? 900 million. I don't know what that means. Sounds an awful lot to me. And I think we're going to be hauled over the coals for this at some point in the future. Cows grazing hard with cows, or sheep even better for grazing hard, they just don't kill a perennial ryegrass or ryegrass clover. They don't do it. It's going to regrow. That's what it does. And cultivation is, of course, the other option. But how do we do that without losing this good stuff? So we bought a shallow plough. This is a 100 horsepower tractor, and it pulls a 5-furrow plough. So it is literally ploughing at 3 to 4 inches deep. And I know 1 or 2 of you have had similar experiences. I met Robert Hyde down in Dorset, who's a good friend, who told me he was using this plough, and we copied what he was doing, and it does seem to do a much better job than the previous one we used. The previous one was going really deep, and I just kept looking at stones, and I thought this is completely wrong. After we ploughed, we power harrow, which makes it even worse. That's probably doing all this damage, Andy, to you. But I don't have a lot of kit. We're a small farm. So I could bring in other people, but I need to do it when the weather's right, and that's the kit I've got. So that's what I've done. But I do get, and please, when you're sowing seeds, it's the one thing I do know about, especially with small seeds, they nearly always need a good seed bed. Bigger seeds are slightly different. You can rough them in a bit. But when you're sowing lays, particularly, some of these seeds are like a millimetre in size. They're no good in a rough seed bed. We can't get away with it with those. So you do need sometimes to cultivate. So I'm just saying, there's horses for courses, and cultivation is no bad thing when you're sowing small seeds. Richard Gantlett, I don't know whether you're in the room. Richard Gantlett is running Yatesbury House Farm in Wiltshire, and I've known Richard for at least 25 years. And he was telling me about this system that he had developed down there in Wiltshire, not the same soil type, but fairly light stuff, really. And he rips the surface real shallow, and probably 3 or 4 times to kill his lay. And this is midway through the process, and you can see that it's going to need cultivating again. But a couple of things here. You don't need a skilled ploughman to do this. I mean, how many in the room can plough really well? I bet not many these days, because they're not doing it so much. But this is a job, and with greatest respect to students, but if you're on student placement, you could do this job. Once it's set, you just keep going. And it is shallow, and it doesn't seem to me to be doing the damage at any depth. In my eyes. There's questions, of course, around weed control. If you go 3 or 4 times, this is a bastard fallow, really, so you need dry weather. This would be a good summer to do it, wouldn't it? But can you even break the surface? But it would be a good summer to do it. And if you don't do it this way and you shallow plough, you might get plants like chicory coming up after the lay. And you all know, if you've got herbal lays in the rotation, chicory is, the good and the bad boy. It can be a pain in the arse when it's in a cereal crop. And if you can't kill it, you get it with 4 litres of Roundup. You wouldn't get it with a cultivation necessarily. But I tolerate it. I'm very happy. I don't need a perfectly clean field. And regenerative farmers seem to get that bit. Other farmers, I've been trained to do the blueprint, monoculture, perfect system, but I don't mind a few weeds in the crop if they're not doing any harm. I don't mind at all. And so we've done this now for only 1 year, and when Andy does the next measurements in 12 months' time, I'm hoping that we're going to see some difference. So we've basically divided the plots now, and we do one with a shallow plough and one with a minimum till, and we have a Vibraflex, 2 Vibraflex, one with a little narrow tine on it and another one with a duckfoot or goosefoot tine. So it's really shallow, and it does feel very different, and I don't know yet which is going to work or not. So I would suggest to this room in particular, unless you're organic or biodynamic, I would suggest that you keep all options open. But I just don't think we should be relying on any one. And I'm really pointing the finger at glyphosate because, we still need all the tools in our toolbox. And I'll give you one small example. As I left the farm last night, our control plot of wheat has got wild oats in it and blackgrass. Now, the graminicides that we're putting on that are just not doing the job. So what am I going to do? What would a farmer do? Well, you'd probably plough it. And so even though we've had 3 years of no-till, I'm sorry, Andy, on this occasion, we're going to plough that bit up, which means the experiment won't be consistent except for the monoculture crop we grow on it. So,
but you now know what you need to fix.
Yeah, I found it incredibly valuable to be critiqued with good science, looking at things that I was unaware of. So thank you very much, because it's really, I tell you, I don't sleep at night anymore.
Oh, that's a pleasure.
So I'm always trying to get away from glyphosate, and one of my favourites to destroy a cover crop is I'll wait till it's -4 and below. I get up at 3 o'clock in the morning and I go and destroy that cover crop with a Cambridge roll, which worked fantastically well for me. I can normally achieve 90 to 95% control as long as I've got a good cover crop there. But that's all back to our maritime climate. If I can establish a good cover crop, I could step away from glyphosate. It works really well, but with the weather conditions we have, I can't always guarantee that good cover crop. The other way that I do it is using a crimper roller. I tried the standard crimper rollers, and what I found with the rigid 3-metre roll, one side of the roll would be chopping the cover crop up, which isn't crimping it at all. The other side wouldn't be doing anything at all. You think your fields are flat, but they're not. So working with Trevor Tappin at TT Engineering up there, he built me, I told him what I wanted and we had loads and loads of phone calls. He built me a crimper roller, so it's 3 individual metre rolls that follow the contours of the ground. And then we've also got it on an air pillow so we can set the pressure. I could set it that light that I could run it across your foot and it would just tickle it, or I could set it so hard that it would break every bone in the foot. I can set the crimper roller for the crop that I'm trying to terminate, which again, if I can grow a good cover crop, it works really well. Because if I was doing a talk in the south of the country, there'd always be somebody at the back of the room that would put their hand up and say, we don't get frost down here. Well, I don't always get frost, so I can use the crimper roller. The two together work hand in hand. And again, if I haven't got 100% kill, I can still go in with glyphosate, but at a very low rate. So I'm keeping all my soil biology working and doing all the work that I want it to do. It doesn't like disturbance. I always class ploughing as somebody coming into your house and knocking your house down with a ball and chain. It's so aggressive. Fungi don't like disturbance. And just as you start to lay a few bricks, somebody comes in with a ball and chain again. It can't get established. So I'm always looking to step away from inversive tillage. I'm not against ploughing. If it's a tool, if we need that tool, let's use it. It's moving it forward. My goal is always to keep that soil working and moving it forward. Profit is always my number one consideration, because if we're not in profit, it's a waste of time, because there's always somebody ready to take your farm on. So making money has always got to be my number one priority. But soil health is a close second, because if you've got a healthy working soil, you've got a profitable business, in my experience.
So there we are. Thank you both very much. I hope what we've tried to convey, not yet. I hope what we've tried to convey is that there should be no one way to farm, that there are lots of ways you need to juggle depending upon circumstance and context, and that how you move between one part of the rotation and the next is probably the really critical bit we need to collectively, farmers and scientists, need to understand how to do as effectively as possible. So we should have a little bit of time left for you to participate in this discussion now. So I'd be very keen to hear your experiences, how they relate to what we have been talking about, or how they may be very different, or how you may see things very differently to the way we've just described. So there's a roving mic moving around soon. So please, if you have any questions, let us have them. The lady over here is doing it.
Are we on? Just a question for the panel. How can you effectively crimp a multi-species cover crop, or should you move to a single species?
In my experience, as long as that cover crop has all reached the flowering stage, I can crimp it. It works 100%, but it's got to be at the flowering stage, otherwise it just regenerates, which all comes back to, if we can have the right weather, Harry, then I can terminate with a crimper roller every single time. And the beauty I found with that, with these dry periods, if I terminated the cover crop with grazing animals, even though I'd only ever graze 2/3, I'd always leave a third. It would still open the crop up, so my soils were warming up a lot faster. They were drying out a lot faster. So my yield of spring beans would only be about 2.5 tonne to the hectare. Where I'd used my crimper roller and I'd kept that soil covered, it was a nice thatch all across the soil, I was doing 5.5 tonne to the hectare. So that robbing grazier wasn't paying me enough for terminating my cover crops. And that's where I know animals are always, and I'm not against grazing, I like animals. I'm not a stock farmer anymore, I used to be. But as an arable farmer, they can take an awful lot of nutrition off the farm and nobody's paying me for that. whereby me terminating and keeping that cover on farm, I'm making more money and I'm keeping all that nutrition on farm, because I did sit down and work out how much nutrition that animal was taking off farm with offtake, and it's quite frightening, and no grazier is ever going to pay me the money I think it's worth. So I don't mind doing it, grazing. I'm not against it. If it's going to enhance the crop, I'm all for it. But at the same time, if it's going to reduce my profit, I'm all against it.
Briefly to add to that, I think you can use them to kill, but it's, in my experience, hasn't been reliable enough, and especially with multi-species cover crops, definitely doesn't work with perennial crops like grasslays. But I think if you get a frost, I guess Gabe Brown's answer would be the same thing. I mean, he's so well known for it, wasn't it, at the beginning? But it hasn't caught on. I mean, we've made these crimper rollers, but you'd all have one now if they worked. And we haven't really seen uptake of them, have we? And I think that tells you a lot.
Tim, just with the crimper, our experience of crimping a cover crop was that we found it failed if the cover crop wasn't tall enough, because you weren't getting enough bites on the stem. But I noticed, I thought that your teeth were quite close in that photo, so presumably...
That's why it was one I had made. My teeth are a lot closer. I'd like to get 3 or 4 crimps on the plant going up the stem.
Because that wasn't a very tall cover crop, but
No, it was more just for show. I'd like them. My cover crops are normally up here.
And can I just ask, going back to the start, the photo of the lupins that you'd drilled into a cover crop, can you just talk us through what that cover crop was and when you established it?
It was a King's Super 10 mix, which I get on with really well. It's 8 different family species in there, which is what I'm always after in a cover crop. It was established early August that year, so it was a really good cover crop. And then somebody called Neal wanted me to plough that in. It was just disgusting. But it just showed it was really, I learned an awful lot from the trial. It cost us money because, to say, the bit where I'd done the cultivation, I lost money on that, which probably the first time in my career I've lost money on a crop. But it was still valuable. I learned a lot from it, and it just, to me, the amount of weeds that came from disturbing that soil, and soil is always trying to repair itself. It's always about being the observer and seeing what that soil is telling you, and the amount of weeds, the damage I'd done from that cultivation far outweighed the damage I did with applying a bit of glyphosate, where my soils regenerated that and broke that glyphosate down. To me, it was hands down, the glyphosate is the better option, but crimper rolling would still be my number one. But it still had glyphosate on top of that crimper rolling because there was some little weeds in the bottom, so it had a low dose just to tidy it up, so I'd got a clean crop.
Tim, can I ask you, does that low dose mean that we're more likely to get resistance? So if you had a ryegrass, like we've got Italian ryegrass resistance now, would a low dose make that more likely?
Not because I'm using fulvic acid, I'm getting more absorption into the plant, and that's what it's listening to what I'm saying, because I've got that cation exchange capacity in that fulvic acid that allows me to put a low dose on. If I wasn't using that, it would still have to be a higher dose. But it's using the best of the bits that we've got to come up with a solution that's going to limit the amount of damage.
Hi. I've got a question for Andy about specifically the low pH response that you saw, the stress response. If we're acidifying around the root zone, so when we're drilling with citric acid, for example, what amount is too much to sort of elicit that stress response? Should we be worried about that?
Well, the response we were looking at there was not a sort of, it wasn't a life-threatening response. It was a natural response that microbes effectively replace the lipids with more structurally robust ones to make sure that all the proteins they hold in their membranes, which are carrying things into the cell and carrying things out of the cell, that they still function. So that's not, we weren't seeing was death there, we were just seeing a response, but it does tell us that things are changing. And although we do know that certain microbial species have preferences for pH, and so, yes, if you're changing the pH in the rhizosphere, you would expect to see a change. I don't think that's necessarily a bad thing. That, to me, is a resilient system responding to an environmental change. So, and there was nothing unusual about your pH, 6 to 8. It wasn't like it was a pH 3 soil that was really toxic. So, no, I think what we're seeing is a natural response of a healthy environment responding. So that's how I would interpret that. Thank you.
Hi, this is question relating to the crimp rolling and maintenance of moisture in the spring and wanting to keep that moisture. I was just wondering how that differs depending on different soil types, as we're on quite heavy soils and we're wanting the soils to dry out so we can get on early and know that we're not going to run straight from being wet soils into a drought period. So I was just wanting to get your thoughts on that balance.
Everybody's looking at me. It's always a difficult one. It's if you've got that crystal ball and you know exactly what spring you're going to have, that it's an easy decision, but you still never know, because it's like any cover crop. If you've grown a fantastic cover crop and you're really proud of it, and then all of a sudden it becomes your worst enemy because the soil underneath is cold, it's wet, and you can't get a crop established. So it's what I do, the way I do it, I'm always monitoring the long-term forecasts, and I'll make a decision at some point of what I'm going to do, and I can only act on the best information I've got on that day that I make a decision. If it's the wrong decision, I'll try and counteract it in whichever way I can. But that's farming. It's never easy. I'm not answering your question probably, but there isn't an answer. It's every individual farm is different, and you've got to decide earlier on in the season what approach you're going to take for it. But when it works and you get it right, it works bloody well. But if I was on a heavy soil and I knew it was going to be cold, then I'd probably have a different approach, because it's not going to warm up underneath that thatch, and then you're going to have a problem.
Do we have any more questions? Oh, yes. Just down here.
Put your hand up so I know where to go. Oh, sorry.
Hi. Just a quick question on strip till. I know it wasn't in the presentation, but would be interesting. Strip tillage.
I've not any direct experience with it. Great concept. Tim?
I started off strip tilling, which quite a lot of direct drillers do. I had good success with strip tilling, but I still felt I was moving too much soil for my soils. But it all depends on what health your soil's at and where you are. If you've got a soil that's very anaerobic and compacted, strip tilling is a really good way to start and make that soil and step away from all the addictions of being a drug addict, of wanting all these synthetic inputs. So it's doing it gradually. You can't afford to go cold turkey because you'll be bankrupt overnight. So strip tilling, yeah, it's a very good introduction, but it all depends what you're dealing with at the start of the journey, in my opinion, because I wouldn't go back to strip tilling now because my soils are in such good nick. It would be nearly as bad as Andy asking me to plough it. It's...
Are you ever going to get... What happens if you get a cheap plough come up next door? You think, oh, I'll have it for that odd occasion where I might want to dry my soils out.
I haven't had a plough for 11 years, and there's no way I'd be looking to buy one again. And I love ploughing, don't get me wrong. Once
So again, just on the roller crimper, I imagine that you're waiting until spring, are you, sort of later spring, until it's getting a bit taller so you can go and crush it with the roller crimper. Does that not mean it's then too late in the season to plant a spring crop?
No, it works really well for me because if I've grown a good cover, which I can crimp, then it's starting to senesce anyway, so that crimper roller just finishes it off. It's always a dilemma. Do I roll? If I've got the frost, do I roll or do I wait for crimping? And that's another decision. I'm looking at the long range and trying to make a decision. I don't always get it right. I'd be lying if I said I did. But if I've got a really good period of frost, I'd use the Cambridge roll because it works so well. And I'd leave that soil covered then because all that debris is on the top. So it's done the job for me. But if we don't get the frost, the crimper roller there is backing me up. But if I could already start to see what I thought was a pattern of a dry year, which this year I said in February it was going to be like this, and a big head now I could say, oh, I got it right, but I could have easily got it wrong. It's but the crimper roller comes into its own then, and that's, yeah, it's, we're always thinking on our feet. That's what farming is, in my opinion. And farming this way, we're dealing with a living entity, so we've always got to be observing and thinking. Chemical farming's easy. I could do that from the farm office, looking at the calendar and looking out the window. I wouldn't be far wrong. That's how easy it is to farm this way. You've got to think. It's not easy.
Can I ask a question? There's a question. Can I ask two questions there? One, before we go to the lady in the front, can we just ask the gentleman, Jed Solomon, behind, just to comment? Jed Solomon is a PhD student at Oxford University and part of the CHC project. I know because he's doing work on our farm. But could you talk about the healthy soil's activity and its ability to respond after damage with either glyphosate or plough?
Yeah, no, thank you very much. And this is something we've been looking at in quite a lot of detail at FarmEd specifically, and understanding the impact across the entire soil biology. So looking at all of the larger things all the way down to the microbes too. And actually it's really about building this resilience that I think Andy was talking about previously, about how we think about our soils and how they are able to respond to lots of different types of disturbances. So disturbance is something that we utilize. It's a concept in ecology that is sort of very common, but I don't think we really think about it a lot in farming. And actually there's good disturbance and there's quite catastrophic disturbances as well. And you always want to make sure that you're stimulating a system with good disturbances, because really that's what creates the complexity and the idea that and sort of stops the system succeeding into something else that could possibly be a less sort of complex state. We really want that complexity and the diversity in all of the life in order to be able to provide us with a lot of different types of functions, but not even just functions that will be active in any one moment, functions that can be active if there were to be some sort of shock that were to come into the system. So for example, that shock might be a drought, it might be, for example, glyphosate, it might be a deep plough, but you want to be able to sort of create the resilience within your soil biology that is able to respond to those shocks. So if a specific actor, let's say a specific organism, is knocked out for whatever purpose, there is another organism that is able to come in there straight away and fulfill that function. So it's really important that we're trying to think about our soil biology as sort of the mediating factor between all of the physical and the chemical components of the soil. Over time, that biology can really do wonders, but it really does take time. So I think a lot of the interventions that we've been speaking of today are really fantastic ones that can help solve some of the short-term issues that we have on our farms. But really, we want to be thinking about in the longer term how we can support all of that soil biodiversity and make sure that it can give us a complete range of functions that can respond to all different types of situations and conditions.
Nice one.
Hi, I'm Gina from Sustain. Thank you so much for an interesting and honest panel discussion about this. My question is what the farmers on the panel think. Do you think that the government is doing enough to help farmers become less dependent on glyphosate? And if not, what do you think they should be doing better?
Is it the government's problem or is it our problem? I think it's our problem to solve. I haven't got faith in government. Change always comes from people. We don't want to be eating glyphosate. We don't want it in our food system. We've got to come up with a system that's far more resilient, that we can farm without it. And I think, I'm happy with the way I'm farming now. No way I'd desiccate a crop with it that's going into the food system. So I think it's farmers and exchanging knowledge and moving forward and helping each other. That's my opinion.
So, yeah, I think it's a very good question, and I agree with Tim. I would add also, I think we are on a journey. Regenerative farming is a journey, and it isn't, as somebody said just now in the audience, you don't want a cliff edge, because that is really bad. You won't have an income if you stop doing what you're doing now. So the transition's the important bit, and I think if it's not the government, which, let's face it, the government's already putting money into sustainable farming incentives, but if you're looking requiring more money for a transition over a rotation or more, that has to come from all of us collectively somehow, because the benefits of a healthy food system, of course, clearly massively outweigh just one or two small items. So, for example, my view would be, if you had a healthy soil, you know what I'm going to say, it's healthy soil, healthy food, healthy people. And it's so true, and it fixes everything at source. It's apolitical, everyone would agree with this. So I think the money is really important. It needs to flow. And I think we need, as a crowd, we need to make it flow through the food system. I know it's difficult to see a different system ahead of us. It's almost impossible to imagine. But look, I'm 62, and there wasn't Amazon. There was no internet. There was no iPhones. Electric cars, they didn't. I mean, all these things are happening. Change is ahead of us. We're moving into completely different times. And we're going to struggle if we carry on doing the same thing. So I think government and the eaters of the food that we supply are as important as each other. Oh, sorry. I just have one other thing. If we're not careful, we're going to have the 8th principle of regen farming. So you know what the 5 are. Don't need to repeat those in this room. I've written the other 2 down with one I learned today. Context, focus. Is glyphosate use of going to be the Achilles heel of regenerative agriculture? Because I bet you if you asked the eaters of food what they thought about that, they wouldn't be overly keen on it, like we've been discussing in this room today. So, yeah, transition, more resilient, like Jed said, that surely is where we're at.
Got time for one more, and then I've got a question I want to ask you all. So,
hello.
Perhaps you can come up and ask yours personally later. Yeah? Okay. Thanks. Sorry, go ahead.
Thank you. Just a very quick, it's more of a summary of your message, really. I guess basically in this room, it's all very well understood, potential health impacts of using glyphosate, etc., and, the need to reduce reliance on it. Do you think... Is that going to be mainstream? Because working with farmers is a huge range of understanding of that. And how can we speed up that process of getting that message out to the more mainstream farmers?
The one thing I've learned on my journey, you can't change people overnight. If you push them too hard, they go a couple of steps and then they go 20 steps back, and it's a couple of years before they come again. The main thing is we keep giving the knowledge out there in the hope that that seed that you've implanted in somebody's brain starts to work. And the more proof that we can give that, the more people start to change. And I think if you can start to show it on farm, because using glyphosate on a really compacted anaerobic soil, that glyphosate lies there for bloody ages. And that's where you can have the following crop taking that glyphosate up. It's going into the food system again, and we've got a problem. But my way is it's knowledge. Once the light comes on, you can't switch the light off. But getting it to come on can take a few years. And I think the best example I've ever had of that is I did a farm walk in 2012, and one guy on the farm walk just kept giving me hassle the whole way around the farm. In 2019, he phoned me up and apologised because the light had come on, and he knew he just made a total fool of himself. But at the time, he totally believed what he was saying to me. But it's just reaffirming that knowledge and passing it on is how we're going to make people change. You can't force it.
So I'm going to draw this to a close. Thank you very much for all your questions and engaging. But before you do go, I'd just like to have a show of hands to sense how many people in here today are feeling threatened by the possibility that glyphosate could be banned at some point. Okay? No, threatened by the fact that it may become a tool that you're no longer able to use. I say there's a reasonable amount of people that are threatened by that. And so we have about 8 years to try and come up with a solution that works.
One rotation.
Okay. Thank you all very much. And if there's questions we haven't answered, I'm sure we'll be happy to answer them. Thank you.